Hello, everybody. So I'd like to thank you for being here and giving me the opportunity to show you this data. So as we've been hearing in this session, immunotherapy has proven clinical efficacy and tremendous potential in a number of cancers. But even patients who experience clinical benefit often achieve only a partial response. So such that additional work is required to improve immunotherapeutic strategies. We need to have a better understanding about the dynamic interactions between the tumor microenvironment and the tumor-infiltrating leukocytes. And also we need to understand the effects that these therapies have on tumor-infiltrating leukocyte biology. Syngeneic mouse tumor models, they're extremely useful tools to study immune responses inside the tumor because they have a fully competent immune repertoire. However, the phenotypic and functional analysis of these T cells is extremely challenging from a technical point of view, and they're very labor intensive. So, as this figure illustrates here, solid tumors are complex tissues, and they contain multiple cell types, and the composition of these tissues and the amount of tumor-infiltrating leukocytes is highly variable, which complicates the analysis of these individual populations. Flow cytometry is an extremely effective way to analyze immune cell populations. These cells can be characterized according to the expression of different, and identified and characterized according to the expression of different molecules. However, as this very busy slide shows, the analysis of these tumor-infiltrating leukocyte populations can be very challenging, because the frequency of leukocytes inside the tumor can be extremely low. And this, in particular, poses a problem for small populations that might escape analysis as they can get lost in the background noise. Taking this as an example, the B16F10 model, this is a transplantable mouse melanoma cell line that can be transplanted subcutaneously into mice. And for example, 1 to 2 weeks after tumor inoculation of 500,000 the B16F10 melanoma cells subcutaneously, you get tumors that range from 200 to 500 milligrams per mouse. After dissociation of these tumors, you get between 10 and 20 million cells in the tumor. Now, taking the example that I just showed you, of those, only 2% are leukocytes. Of those, only 10% express CD3, and of those, only 16% are CD8 T cells, which means that if you want to analyze 100 events of a population of 5% of the CD8 T cells, as for example antigen-specific cells, you would have to analyze over 6 million total T cells, total tumor cells, which means that running the FACS at 4,000 events per second, you would take 26 minutes just to analyze one sample. And if you have 9 samples, for example, which is a typical 3-group with 3 replicates experiment, this would take you over 4 hours just analyzing the cells. So it's fundamental to use effective tools to streamline this workflow and to be able to generate some high-quality data. So to this end, we have developed solutions for all steps of mainstream cancer research and established workflows that combine tumor dissociation and cell isolation to decrease the time required and increase the quality of the data generated. And today I would like to show you some of these solutions that can dramatically enhance your typical cancer research workflow. So this, of course, starts with collecting and storing the tumor tissue. Sometimes when enlarging with large cohorts or using tissues coming from multiple origins or collected at different time points, or for any other reason, these tissues cannot be directly processed. During this storage period, it's important to avoid necrosis and apoptosis, and also it's important to maintain the functional properties and activated status of these cells. Otherwise, we can cause bias on downstream analysis. So for this, we developed the max tissue storage solution that has been developed and optimized for storage of fresh organs and tissue samples. It has been tested for multiple tissue samples, both in mouse and human, and they allow high total yield comparable to fresh tissues, both yield and viability up to 48 hours after collection. And this solution was able to not induce apoptosis or an increase in necrosis, and we didn't see any cell activation or influence in markers expression. We then need to dissociate the tissues to prepare the sample. Solid tissues, such as tumors, they are built up of a mixture of cells, extracellular matrix, basal membranes, and so the goal is to, on one side, disrupt these extracellular matrix and cell adhesion components without destroying the integrity of the cell membrane. But very importantly, the cell surface epitopes should be preserved. Now, usually, a combination of both mechanical and enzymatic treatment is required for efficient dissociation. So mechanical disruption involves mincing, straining of the material through meshes of different pore size, pipetting, etc. And then the material undergoes enzymatic digestion with several enzymes to complete the dissociation. So using the GentleMax dissociator or the GentleMax octo dissociator, the tumor dissociation was automatically and optimized for epitope preservation. So the dissociation was automated and optimized for tissue dissociation. And multiple tissues can be dissociated at the same time, which saves a lot of time. So this ensures a highly reproducible dissociation because it's user independent. Processing time is reduced because mechanical and enzymatic treatment occurs simultaneously in multiple tissues, and the dissociation takes place in a closed system. So by the use of single tubes, we can maintain sterility and high user safety. We generated several protocols and enzyme kits that were optimized for each tissue for epitope preservation, and they were validated in multiple tissues. So once we have a cell suspension, we improve the isolation of tumor-infiltrating leukocytes by developing a new CD45 specific enrichment reagent for magnetic cell sorting based on MAX technology. And this is done directly from the dissociated tumor. Using this technology, the dissociated tumor tissue is directly labeled by the CD45 conjugated microbeads, and then magnetically isolated, resulting in a very efficient depletion of CD45 from the negative fraction and a very high enrichment in the positive fraction. This has been validated in 3 different tumor mouse models, the CT26, B16F10, and 41. And these TILs were purified were isolated to purities over 80% and with very high recovery. As a consequence, before the tumor isolation, the cells of interest were vastly outnumbered. When 100,000 cells were acquired by FACS, in this example, the analysis of the subpopulations was pretty challenging. You barely see any of those subpopulations. However, when the same number, 100,000 cells, were of the enriched fraction were analyzed, this was enough to have a much better view and analysis of the different populations. In practical terms, this isolation of TILs results in a massive fold increase of numbers of cells of interest analyzed if we collect the same total number of cells. And this translates in significant increase in the quality of the data. So if we go back to the first example that I showed, this is the kind of data that we are able to generate using when we analyze 100,000 cells of the total tumor cell suspension before isolation, and then after isolation in the tumor fraction, we can now have a much better picture of the immune component in the tumor. So if we go back to the same type of analysis in the beginning, so it translates into significant time savings if the number of cells of interest is kept constant. So we can either take 4 hours for our complete analysis of 100 events, or down to 5 minutes. This significantly improved workflow can be further streamlined by using the Multimax, which allows the parallel isolation of up to 24 different samples, or the fully automated Multimax X, which is a fully automated walk-away solution with liquid handling and high-throughput cell high-throughput cell separation. All 3 solutions were validated for isolation of TILs, and they all have extremely high purity and yield. So in summary, I showed you here that how we can streamline several components of the cancer immunology research workflow. I showed you a solution for tissue storage and shipping that has been optimized for storage of fresh organs and tissue samples. Automated tissue dissociation that allows highly efficient and reproducible dissociation with automated parallel mechanical and enzymatic dissociation that was optimized for epitope preservation, and a strategy for magnetic enrichment of tumor-infiltrating leukocytes, which allow enrichment of CD45 positive cells directly from the dissociated tumor tissue. This allows reduced time and cost of downstream analysis and greatly increases the quality of the data obtained. So I'd like to finish by acknowledging my colleagues at Miltenyi that were involved in producing these data and our collaborations. And I'd answer any question.